Good morning. I am Young-ho Kim from Tulgen. I am very excited to be here. I am very honored to be here. I am the chairperson of Technology Session 14, titled The Future of Genome Editing Technology, in part of the BIOPLUS-INTERPHEX Korea 2022, shortly BIX 2022. This morning, you guys come over here at COEX through hot and humid summer air. The theme of BIX 2022 is Summer in BIX. The organizer of BIX 2022 want you enjoy the conference such as summer vacation, I think. Thank you very much for taking your precious time to COEX despite your busy schedules. Okay, now let's start the session in earnest. I briefly introduce in this session. We will mainly talk about CRISPR Cas system. The CRISPR gene editing technology has been awarded the Nobel Prize for its breakthrough technology compared to the existing genome editing technology. This is the spotlight technique used by any lab that wants to edit the genome. In 2012, the US Patent and Trademark Office followed the principle of invention priority, and there were patent dispute between various organizations that filed patent application at the time. From life science research and medicine to genetic engineering, how far can this technology change and how far can it go? Learn about the present and future of CRISPR technology in our The Future of Genome Editing Technology session. Please enjoy. Okay, now I'm introduce the Tulgen business model to you. Actually, we announced the vision of our Tulgen year 2017. Our slogan is Innovate Genome. That means we revolutionize the genome editing technology and for the human therapeutics and agriculture area. Actually, Tulgen has tools for read the genome and edit the genome. The year 1999, the Tulgen founded, and since then, more 23 years passed. Tulgen developed zinc finger nuclease and TALEN, as well as CRISPR Cas9 systems. We wanted to make people's better health and happier life. You already know the engineered nuclease called can targeted cleavage of our genome sequence and then make a knockout or repair and insert the genes into the target site. And then we can make human therapeutics. Also, we can improve the phenotype of agricultural products. Or many labs use those technology for R&D. And also, it can be used to the industrial biotechnology. This slide shows Tulgen's business scopes. Tulgen has a platform technology for the CRISPR Cas9. We filed patent in 2012, and then after that, we improved our platform technology, and also we made improving genome editing tools. Also, we made several R&D programs, and then we patent also. That means our business area make diverse to the platform, our licensing area or human therapeutics area. Also, we did agricultural areas. This slide shows a patent landscape of CRISPR gene editing technology. This morning, Professor Sixnis showed some CRISPR technology developed. And actually, in the side of company areas, the patent application of patent is very much important for the business. In the left side, you see the University of Vilnius, the Professor Sixnis in there. And this is CVC group. It means UC Berkeley and University of Vienna and Emmanuel Charpentier group is called the CVC group. They published patent in 2012, early 2012. But these patents mainly in vitro or prokaryotic systems, CRISPR system. And the right side of this slide showed this CRISPR Cas9 system can be used to eukaryotic genome editing. This is a system patent. This is the method patent. And you see the toolgen is most earlier application of patent for the eukaryotic systems. toolgen published 2012 October 23 is the most earlier time, and then Sigma is December 6 and Broad December 12. That means toolgen is a most higher position for the IP, the platform IPs. And we did application and published patent to all around the world, and we have strong CRISPR IP portfolio with issued patent in 9 countries, almost over 20 published patent in the world. And this slide showed what groups can make a CRISPR technology to the develops. And you see here the Doudna and Charpentier group, and this is a toolgen group with Professor Jin-Soo Kim, and this is Feng Zhang group, has a key inventor of CRISPR Cas9. And CVC group has a patent, system patent, and then they out-licensed the CRISPR IP to Intellia, Caribou, and Corteva CRISPR Therapeutics ERS companies that they make human therapeutics or agricultural products. In the case of toolgen, we solely own our IP, and I said the world-only company with right in all business area. That's the toolgen in South Korea. For the Broad Institute, they out-licensed to the CRISPR IP to Editas Medicine. They made human therapeutics using CRISPR IP. And I mentioned earlier, toolgen founded in 1999, and at that time we deal with zinc finger nuclease for the genome editing. At that time, the worldwide only 2 companies using zinc finger nuclease for the genome editing area, toolgen and Sangamos. And year 2011, Talent is most valuable tools for the genome editing areas. At that time, toolgen and Selectis can be made some therapeutic technology using the Talent. And Calix made some useful agricultural products using Talent. And year 2012, the CRISPR Cas9 is come up with us, and then a lot of companies using the CRISPR Cas9 technology for the human therapeutic development and agricultural product. You see here the Toolgen, Editas, CRISPR, Intelia, something like that. And our business strategy is we have genome editing platform technology, and also we improved those technologies to the more practical. And then we can out-license our platform IP to the interest group. Already we out-licensed our IP to the 18 companies. It's a Monsanto, Thermo Fisher, Kijin, like that. And then we also contact with another companies. They interest our IP. Also, we developed the therapeutic asset and agricultural asset using our CRISPR IP. And then this product in therapeutics and agriculture, we can out-license those products, or we own product, our agricultural product in the future. And this slide shows our therapeutics area, genome editing therapeutics area, and genome editing crops and genome editing animals. Okay, I just skipped this slide because Professor Sixnis already showed the CRISPR Cas9 systems. And we didn't stop the original IP. We improved the original Cas9 because we wanted to make a more specific, more active Cas9 make to the world. One of the famous, the very specific Cas9 we named Sniper Cas9. We already make. And also, the Professor Sixnis told us the size limitation of Cas9 to deliver the in vivo. And we select the CJ Cas9. That's the half size of original Cas9. We got the CJ Cas9, and then this CJ Cas9 can be into the in vivo gene delivery systems. Okay, in the case of human therapeutic area, we wanted to make license out as soon as possible because most of active technology transfers is in the preclinical stage. That means we must out-license our product to the global biotech company, global therapeutic company, and then we got the money, and then we reinvestment to the R&D, and then we made another pipeline, and then we out-license. This is a kind of virtuous circles. This slide shows briefly our therapeutic pipelines. We have almost 10 pipelines actively, and this is the in the CNS and PNS area therapeutic program. It's a CMT1A and PMD disease. That's the genetic disease area. Especially CMT1A, we collaborate with CMT Association in US. And also we have the eye disease called wet AMD therapeutic area. We developed some assets. And also in liver disease, there's two main programs. One is hemophilia, that's the genetic disease. And the other is HBV, it's an infectious disease. we now the animal efficacy data. And also, we have ex vivo cell therapy area. We make a genome-edited CAR-T, and then very much improved their activity in CAR-T area. And also, we did genome-edited MSC, the stem cells. Okay, we are now deal with our CRISPR Cas9 system delivered to human. We are making the AAV delivery systems, and we select several serotype of AAV can be into the neuronal system or liver or eye. I would like to more details our therapeutic programs. In the case of CMT1A, this disease is causing the PMP22 duplication is causing for the CMT1A. We downregulate PMP22 expression using our CRISPR-Cas technology. And this is some animal efficacy data. When we apply our CRISPR-Cas9 systems, this disease model mouse can be restored. This myelin is restoring, and the behavior is restored. Also, we did some specificity study. This means that nowadays the FDA warning some off-target activity, but we found our CRISPR-Cas9 system, we select some target guide RNA, cannot digest any other sequence, only PMP22 area. Also, this is wet AMD. We have deal with the therapeutic area. We make a CRISPR-Cas9 targeted to the HIF1 alpha or VEGF1. That means antagonizing angiogenesis. And then we did some result from animal study, and you see this phenotype change after the CRISPR-Cas9 introduced. Also, we have liver disease asset. In the case of hemophilia, we select some genome site, pinpoint this genome site, can make knock-in with other the wild-type genes into this area, and then this area can very safe. We can call it the safe harbors. And then if we knock in a gene into this area, the gene is more about 200 times expressed. That means we only need small amount of genes knocking into that area, and then we can get the phenotype. This is another platform technology, and then apply more the therapeutic protein replacement study. Also, we have ex vivo programs. We now deal with immune cells, mesenchymal stem cells, and hematopoietic stem cells. We did genome-edited cells, and then we improved their functional activity and survival of the stem cells, such like that. This ex vivo program, we deal with the collaboration base. In the case of immune cells, we did genome-edited CAR-T. Collaborate with Caserix. This Caserix is an Australian company, and then we have a nice interaction, and we have a great result from collaborations. Also, Genexine, we did collaborate with Genexine, and then immune cells developed. Also, the mesenchymal stem cells, the Vivasome or TNR BioFab, we collaborate with develop more valuable mesenchymal stem cells using genome editing technology. Okay, this slide shows some results from genome-edited CAR-T. We call it next generation CAR-T. This is recurrent models, and if we treat our next generation CAR-T into the mouse, this mouse cannot be recurrent the cancers. This is the commercially available CD19 CAR-T is recurrent after week 6, but our next generation CAR-T is never come back the cancer cells. It's a very astonishing result that can make collaborate, and then actually we hope this year or early next year we get into the clinical stage. And now, okay, I just move to the last one. This is just agricultural example of our genome editing areas. And we did make genome editing soybean, genome editing potatoes, something like that. Okay, we want to expand our collaboration and partnership in agriculture or livestock field. These companies in Korea or outside Korea, we work with those companies and institutes. We can make some improvement. This is our company's leadership. You can see our homepage. Okay, now time's up. Okay, if you have some questions, raise your hand and the staff deliver the microphone to you. Any questions? Okay. Now let's move to the next. Okay. So Monica is waiting for us. Just introduce yourself.
Yes.
Okay.
Okay. So hello to everybody. So I'm Monica Paule, the CEO of Lithuanian company CAS9 that was already mentioned several times by the Professor Shikshnis in his keynote speech. So my goal today is to tell more what CAS9 is doing and how we can collaborate. And you will, for those who, and most of you I think, have listened to the keynote of the professor, so you will see some similar slides, but it's just to show, how all the things are interlinked in the work that we do. Okay, so let's start. So Professor already showed several pictures of Lithuania, of Vilnius that we are coming from. So another one from myself, just to trigger you to think about this country both as a great location for science and genome editing in particular, but also as a great place to visit. And one fact that we are one of those rare cities around the world where you can fly the balloons over the city. So please visit us, and we are very welcoming you, both from science and from more like a tourist perspective. So what CAS9 aims at a very high level. So we are developing new discoveries, innovative applications, and top quality research, in particular in the field of CRISPR-based molecular tools. And by doing this, we want to apply these novel editing solutions that we develop worldwide. So we are fully international company. We work with clients from US to here in Korea, actually. And we really want to create the great, outstanding added value for our clients and partners. So you already know our main founder, Professor Shikshnis. So we founded CAS9 in 2017. And Professor, in his talk, mentioned his previous PhD student, Dr. Giedrius Gasiūnas, who contributed greatly to the initial work with CRISPR Cas9. So he is our Chief Scientific Officer of CAS9. So it's already quite a few years we are in the market, and many, many years to come to do a great work in gene editing field. Yeah, so this is a part of our team, which actually the photo taken a year ago. So we grew quite substantially from that time. And I just wanted to illustrate how much we are into gene editing, and that we even have, art pieces close to the Vilnius University, where professor is located, and the CAS9. We really, as the whole country, love genome editing and know quite a lot about it. Even you can find like QR codes in the public transport that you scan and you can read about, CRISPR and all related things. So we as CAS9 concentrate on certain areas of activities. So first of all, we explore new Cas proteins for various applications. And we do provide research services to help with applying these CRISPR-Cas in the particular field that our clients are working in, or we engage in developing and characterizing totally novel CRISPR-based molecular tools. And here are many ways how we collaborate with other companies, even acting as a R&D partner, delivering all the novel things to our clients. So we are the experts in CRISPR, in protein engineering, and all the related things. And yeah, you've already seen the map that we are really the part of the CRISPR community worldwide. So of course, all of you understand the importance of gene editing, all the essence, all the, how the technology was developing was described by Professor. But what I want to stress that, of course, all the fundamental science has some applications, and in case of gene editing, these applications are really great, covering all kinds of fields that actually impact all aspects of our life. And of course, it's interesting not only from scientific perspective, but also from the society perspective, from the application perspective. So microbiome, diagnostics, all the therapeutic applications, plants, and so on. So CRISPR is actually having a lot of attention, not only from scientific community, from business community, but also from the society as well. That's why I think it's so important that both people like professor from the academic perspective and us as a business people would collaborate and work in actually making an impact with this technology. Yeah, so I will not go into details about the technology itself. I think everything was already said by the professor. And just to stress the importance of CRISPR-Cas, that actually, if you look to the areas, industries that the technology can be applied, and we do work with most of these industries, with the companies from these industries. So, of course, it starts from the research and research tools, agriculture, medicine, diagnostics, and industrial biotech applications. So in more detail, what we see as the topics now, very much in the interest. So apart from the human therapeutics and diagnostics, it's the climate change and food. So we get a lot of, especially from the media, questions. So can CRISPR save the climate change? So the answer is yes, just we have some work to do on that. Like food, can we help the plant species that are getting extinct? Or how can we feed the world? Can we treat the most severe diseases? Yes, we can, and a lot of people and companies are working on that. Can we diagnose the most severe diseases, infections, and so on that we are dealing with very currently? So yes, we can do that with CRISPR. And even to some areas like, can we solve the extinction problem of the species that we already lost, including the animals? So theoretically, yes, and that's actually the great thing that shows to everybody how important the technology is and how many areas are that we can invest in. So coming back to Cas9 and what we do in particular, so we actually help in our clients and partners to solve all those issues that I have just mentioned. So despite of the field, as I mentioned, we work with all different industries. And our approach is to find those best gene editors for a particular application. And we really have a great expertise in that. We have a pool of, first of all, Cas9 orthologs, and later I will tell about the novel type 5s that we are working on. So whatever the application is, we can find the gene editor that could be the basis of that future diagnostic tool, treatment method, plant species, or whatever other application could be. And when we know what's the application, we know what the preferred characteristics, and then we can identify that genome editor. Sorry. And a lot of things that Cas9 does is already published, so you can even look much deeper into the work that we are doing. Yeah, so let's move on. And as I mentioned, we started with the work on CRISPR Cas9, and we have, well, really a big collection of them, but also we are working on novel type 5 nucleases, and the professor already mentioned them and described a lot. So we actually commercialize them, and if you are interested, so definitely you can contact us, because this novel type 5s really have a unique characteristics. First of all, because of the length and suitability to use, first of all, in the therapeutics field. So I will quickly go through just very general examples on what Cas9 was working on. In the field of detection, we were working on the development of rapid COVID-19 test in collaboration with our biggest clinics in Lithuania. The other project what we were doing is detection of single nucleotide mutations without DNA amplification using the chip solutions. So another approach to detection for the gene therapies. We also did quite a few of projects that we can reveal of, because of course in the field of therapeutics, quite a lot of work that we do is quite confidential because of our partnerships and the work that we do with our clients. But in case what we can reveal, so in gene therapies, we worked on expanding the specific targeting, and that's a very important thing that, well, for the therapeutic applications, we worked with delivery using AAV vectors, and also into increasing the efficiency of specific genetic editing with the homology-directed recombination, also all this, as I call, novel techniques like base editing. Also, we have experience with the prime editing, if you are interested in. And summing up, our business model is, first of all, out-licensing. So the orthologs that we have, so the Cas9s, we have a great variety of them, or other novel type ones, even those that we have developed in certain partnerships, we are able to make them available for other fields apart from what we were working with our clients. And of course, the focus is on finding those specific orthologs for the client needs, not just a random orthologs, but really selected for your needs. And we provide the research services, but we really like to call them more like a research partnerships, because it's usually quite intense long-term collaborations that we do. And we can work on adapting our current orthologs, let's say, that you would be interested to license, or engage into development of, well, search and development of novel Cas proteins, and providing the whole spectrum of research services related to CRISPR applications. So we work with partners and clients from many fields. So agriculture, human therapeutics, diagnostics, research tools, and so on. And well, these organizations vary from fast-growing startups to small and medium enterprises, of course, to big corporations, but we do also work with academic institutions and even public sector like clinics and so on to develop CRISPR-based solutions. And just a few names of the companies from different fields. And we have, as a professor already mentioned, a long-term collaboration, very strategic collaboration with the Corteva Agriscience and New England Biolabs, two well-known US companies, but also work, as I said, with the smaller companies and even with companies from Korea, like Ensage, fast-growing startup. And we really think that your industry here in Korea is growing significantly. We see, so many researchers and companies interested in gene editing, and I feel that, you will do a lot of great things. You have a long history of companies working in the field, but also there are many things to do, and we really want to be a part of it because we think that we as countries can really collaborate. We know how to work together, and yesterday we signed the MOU that this collaboration would continue even on the country level. Already approaching to the end. We really have a great team of employees, very experienced in the CRISPR field, and we really get a lot of inquiries to join CAS9 because we're really a great company. Currently, we are close to Vilnius University in the biotech incubator facilities, but we will be moving out next year because we are really growing intensively and already have our super well-equipped labs and office spaces, but are ready to grow even more. And as a company, we really try to spread the word about CRISPR and gene editing, not only on the academic level, but also to the public society. And Lithuania, because of the work that professor is doing for many years, is really recognized as a hub of life sciences. And just to mention, we really have almost 60 years of experience in life sciences as a country. So you would understand that all the infrastructure knowledge that we have, it's really long-term, and we can deliver the top quality research and applications, first of all in genome editing field, but also in life sciences in a broader scale. So summing up, we as CAS9 really offer great opportunities to work on CRISPR-based gene editing, from Cas9 orthologs to all the novel types, and developing these CRISPR-based molecular tools, including applying all the novel gene editing techniques. And we are very open for collaboration, for research development services, joint research projects, licensing, of course, working on a totally new CRISPR applications, and even some advisory if that's what you need. So thank you for your attention. That's where CAS9 is at the moment. And if you have any questions, so please feel free to ask me.
Okay, now we are preparing the panel discussions. Do not leave this room, just wait. Okay, now we are going to start panel discussions. Actually, we prepare some questions to each other. Professor Jung-Hoon Kim make some questions to all. First of all, could you introduce yourself to the audience?
I am Jung Kim. I am working in Seoul National University Hospital. I'm the clinician. I'm the specialist in the inherited retinal disease. Is the disease to the gene therapy and genome editing and cell therapy is the major target for this disease. So that last 16 years I have tried to develop that therapeutics for that inherited retinal disease, small molecule antibody and peptide, and recently I'm more interested in the gene therapy and genome editing because that to help my patients. So that as a clinician scientist, I have some experience about that genome editing, that CRISPR Cas9, and recently I also tried the base editing and the prime editing. Then I would like to ask to the panel that do you think that what is the best genome editing technique, the CRISPR Cas9 or base editing or the prime editing, or even we think that the first generation that also that as a clinician I'm more that the base editing is that the best in my experience. But how about your experience or your expectation for the bit?
Yeah, actually, my answer is so far Cas9 is the best because in clinical trials only Cas9 into that clinical trial. That means the Cas9 is proven technology to the human therapeutics so far. But I'm not sure the base editing or prime editing field is growing bigger, and someday those technology can be introduced to the clinical trials. But so far, Cas9 is best, I think. How about you?
Yeah, so it's always difficult to predict what technology is the best, but I agree with Dr. Kim that actually Cas9 is actually developing now rapidly and advancing to the clinics. So, of course, there are some kind of challenges, and I didn't mention one of the challenges during my talk that actually off-target cleavage is in fact a challenge in human therapeutic applications because Cas9 introduces double-strand break. Yeah. Base editing could just help us to avoid this because actually in the case of base editing, you don't need to introduce double-strand break. The bases are just exchanged, but actually these technologies also have their own challenges too. Yeah.
I think the base editing technology has some problem for the off-target effect. Yeah. So far, base editors do not replace only one base. Yeah. Between the target, there is some mismatches, right?
Yeah. Actually, that in my experience, that still that the base editing have that off-target mutation. But if we usually doing that the gene therapy genome editing, we usually use the AAV. That in my experience, AAV still make that the off-target mutation in the base editing. But when you use that the RMP, that the off-target mutation is so much reduced. But as you say, that I have a concern about that the double-strand break, and then so that the animal experiment is good, the agricultural application is good when I use that CRISPR Cas9. But when I doing the gene therapy, genome editing in human, that kind of the double-stranded break is still we have the major concern as a clinician. So that I want to try to reduce this kind of the concern about the double-stranded break.
Yeah, the selectivity issue is very much important. Also, you said RMP. That means the delivery system is not the AAV,
Yeah.
right? And AAV is a much longer exist in the human cell or tissues that make some off-target effect more. But nowadays we are thinking about the non-viral delivery of Cas9.
Yeah.
Yeah. So move to naturally some other questions. Jong Kim raised the question that it would be good to discuss the prospects of developing non-viral delivery systems. What do you think about the non-viral delivery of Cas9?
Yeah, so I think that actually these delivery technologies are really very, very important in the field of genome editing. And of course, AAV is a preferable vector choice because actually there are already some AAV vectors approved by FDA and then can be directly used in the clinics. But of course, the size is important, and also lifetime of AAV, as Dr. Kim mentioned, is also important. But I would say that actually now we should also look at the LNP technologies, lipid nanoparticle technologies that are developing very fast, and most of them actually experience these technologies during the pandemics because actually we were vaccinated using these technologies. So I think that actually all these kind of different delivery technologies are developing in parallel, and I think that different delivery technologies could be used for different specific applications. For example, for one technology for liver disease, another technology for eye disease, and so on. So it's still a rapidly developing field.
Okay.
And I would jump in with a, let's say, more of a business-oriented approach that there are advantages and disadvantages from the scientific perspective, but in case of a business company that wants to have a product, then it's a combination what works together. So, what kind of method, what kind of delivery, what kind of orthologs and so on. But also, it's not always the goal to have the best results. It's just to have the results that are okay, that are proven in clinics, and also that you have a strong IP position combining, all the process of your application. So that's really sometimes hard to say that one is better than other. It's just actually what works for you and what enables your business that you want to do with the CRISPR.
Okay. I'm sorry to the Bongyong-gu. Why don't you introduce yourself?
Yeah, thank you for giving me the turn. My name is Bongyong-gu. I'm currently heading the Center for Genome Engineering at the Institute of Basic Science, which was previously led by Professor Jin-Soo Kim that you must know. I actually have a question that maybe should be, to Professor Sixsmith or Monica. I was truly impressed today that you have a great number of Cas9 and related proteins. It's truly a plethora of Cas9 type. This also means that we now have so many options. But there was a little bit, a limit of information which of these Cas9 or related proteins have a certain special functions, because I know that there are some other variants that from different species that we are using to replace SPCAS9. But what I have been reading from reviews is that SPCAS9 seems to be still the best Cas9 to be utilized. I was wondering whether you have some better forms that you can think about other than the size, because today you have emphasized the smaller size, but I was wondering whether you have something that has better immunogenicity or better stability or robustness. Also, something that has a much better specificity by having longer recognition sites. So this could be a very nice characteristic of all these different Cas9 variants. So I was wondering how you vision into this and how your company can migrate toward this area.
Yeah, so it's a good question and a good topic for discussion, because of course when you have such a variety of different Cas9 orthologs, you would be clearly interested to apply these orthologs for genome editing in eukaryotic cells. And this is what actually Cas9 is right now doing. So they are trying different orthologs in different eukaryotic cells just to evaluate their efficiency and compare with Cas9. And the preliminary data show that some of the orthologs are as effective as Cas9, SPCAS9, so it's good sign. So some of them are less effective. So, but this is what you expect when you have such a variety of different tools. Yeah, and as you mentioned, it's also important to take into consideration not only the PAM requirements, but also other biochemical properties. And some of these orthologs actually show different cleavage patterns that could be important for genome editing applications, and also different biochemical characteristics like temperature requirements and so on. So that's kind of really a huge bunch of different proteins that could be used for genome editing applications. And I think that, of course, for a company like Cas9, so it's still a small company, it's really very difficult to characterize all these Cas9 orthologs that are available. Monica showed it's probably around 80 now. Yeah, so, and it's really difficult to characterize them all indeed, but actually if you are interested in a particular ortholog, so you can just approach Cas9, and Cas9 will try to develop this ortholog into a genome editing tool for your specific applications.
I think that Dr. Gu pointed out the serious point is that when I doing some experiment using the CRISPR Cas9, I experienced that some immunogenic problem, especially in the inflammatory condition. We injected the CRISPR Cas9 in the AAV. It can make a more serious inflammation. So that I wonder that do you think what is that most less immunogenic Cas9 ortholog in your library?
Yeah. So, of course, there is a problem of immunogenicity with S-pyogenes Cas9 because most of us have already encountered S-pyogenes to some extent because it's somewhere in our nose or throat. And of course, then actually multiple injection with S-pyogenes Cas9 could be immunogenic. And actually looking at the pool of Cas9 orthologs that come from very different bacteria that probably people didn't encounter before could be also a possible solution for immunogenic problems. Yeah.
So for the immunogenicity, I also have a comment. Basically, a smaller size does help because then it reduces the immunogenic peptides. Yeah. And secondly, we have already learned from fluorescence proteins that some fluorescence proteins, such as enhanced GFP, tend to have very little immunogenicity, whereas similar green fluorescence proteins or red fluorescent proteins that are with a similar size but isolated from different species, sometimes they have a very strong immunogenicity, even when we are making them as a transgenic mice, which means that it became endogenous protein, but it can trigger autoimmune disease. So this means that every individual Cas9 protein may need to be tested to see whether immunogenicity is indeed a problem or not. But at the end, to apply this to human, we cannot rely fully on the mouse, or we may need to rely on more humanized mouse or more relevant systems. But since you now have so many Cas9 variants, I think this could be one issue that you can focus on, I hope.
Yeah, thank you. It's a good point.
That means human never met the bacteria that can have Cas9. That's the immuno-free Cas9. You think?
Well, we all know that we have a large number of microbiome in the gut, and the gut somehow knows how to tolerate those. Because we think that some of the proteins will be observed, and there might be also some lesions, which means that these proteins are not always digested to be uptaken. It can be directly get into the body, but still, good microbiome do not cause much problems. So, if some of the Cas9s have been isolated from those gut-living bacteria, there might be a chance to have a very little immunogenicity, I think.
That's a good idea.
Yeah, it's a good idea.
I'm asking to the Dr. Sixnis, Professor Sixnis. How about going back to 2012 or 2010? At that time, the patent related to Cas9 as an application and published it in US or European countries. At that time, what's the mood of researchers using the Cas9 system? I mean, please remember the 2012.
So, as I mentioned during my talk, actually we jumped onto the CRISPR systems very early on. So, after the 2007, when the paper appeared in CRISPR journal, in Science journal, on the first CRISPR demonstration as antiviral defense system. So, and of course, as Dr. Kim, he told that actually you worked before on zinc finger nucleases and talents, probably. So in my lab, we worked before on restriction enzymes. So restriction enzymes are part of the restriction modification system that is another antiviral defense system. And of course, we've had an experience working with these proteins. So then we decided to jump on CRISPR Cas systems. So we were able to purify proteins pretty fast because of our previous experience in the restriction enzyme field. And of course, when we isolated Cas9 proteins, so we just immediately realized that this could be an important tool, because in my previous studies of restriction enzymes, we studied restriction enzyme with the aim to engineer restriction enzyme new specificities. But it was really hard to do, because actually you need to re-engineer protein DNA interface. And actually when we realized that Cas9 is so easy to reprogram, just changing the RNA sequence, so that immediately we realized that this could be an important tool. And of course, we filed in the patent application and then submitted paper to the journal, submitted it to different journals with the same outcomes. They didn't like our paper. Finally, we ended up in PNAS, but other labs were doing much better in publishing, and their papers appeared in different journals a bit earlier.
I think you deserve the Nobel Prize.
Thank you, but this is the decision that Nobel Committee is usually making. Yes.
I can share an episode with you, that because I'm from Vienna, and you know that Professor Shapantia started her CRISPR work from our neighboring institute called Max Parrot Institute. So I heard from my colleagues that when she was there, she actually showed some of her results of her PhD students that the CRISPR system may work as an endonuclease. But when that was presented in our internal the institute-wide seminars, people actually didn't know the value that much. And you also know that Professor Pantia actually had to move from Vienna to other countries to continue her work, and she was also telling me one time that it was really hard for her to carry over all the work while moving around. So this actually means that before something has happened, many people actually do not see the value. So I think now, after seeing 80 different kinds of proteins, maybe we may see the value in a different eyes, but I think there must be some extremely important, some hidden characteristics that we may need to find.
Yeah, but I think that I fully agree with you that actually sometimes difficult to see the value, and this is probably what happened also with our first publication that we sent to Cell Journal. And then the Cell Journal editor rejected it, saying that probably there is not much value in that, but it turned out that actually there is a value, as you mentioned. Yes.
Okay, I'm asking to Professor John Kim. As an ophthalmologist, what kind of medical results are you expecting from gene editing?
Yeah. As you know that the most gene therapy and genome editing usually they applied in the eye, because that variable that monogenic mutation disease occurs in the eye. And then, as you know, that the visual loss is very serious problem. So that the researchers and the clinicians tried to do the gene therapy and genome editing, the most innovative treatment in the eye at the first. So that I think that, as I told you, I have experienced that variable therapeutics, that small molecule and antibody and peptide, all things. Now we are used at the small molecule and antibody peptide is in clinic usually. So that I think that in the future, in near future, in clinic, we will use that the genome editing just like the usual treatment. I think so, because so far we know that the genome editing or gene therapy is that the only the key to help that the patient of with the inherited retinal disease. So that I think that the future is very bright in genome editing the world. I think so, and I think that the genome editing and gene therapy, just that the applied for that just the inherited retinal disease, we maybe that the genome editing would be expanded to the more common eye disease, just like the AMD and diabetic retinopathy and the uveitis. Yeah. But still, we have some obstacle, as I told you, that we should the control the immunogenic problem, the CRISPR Cas9, and we should develop the more the technique. We should make that the HDR efficiency is more high. Anyway, that the genome editing gene therapy is widely used in the eye disease.
You are a clinician with patient, and do you have any good genetic disease in eye for the curing using gene editing technology?
Yeah, actually I'm preparing the IIT that for the retinoschisis, the congenital retinoschisis and the LCA, and this kind of disease, there is no way to cure or treat this disease. It's just that the genome editing or gene therapy is the only way to help our patient.
LCA10 is a target for the Editas medicines.
Yeah.
How about how do you think about the Editas medicines programs?
It's a good way that LCA10 it means that LCA is the variable types. About 18 types is there, and then in my the patient when the investigation of the genetic mutation in my patient, the genetic mutation is not 18 types. It is more than, so that we need more the variable the treatment option. LCA10 and the clinical trial in Editas is a very reasonable and very good. But problem is that the LCA10 is patient is not so many, so that it is just that only small number of the patients can be helped by that treatment. So that we need another treatment for that LCA1, LCA2, LCA3, and LCA4. Yeah.
Yeah, that's very tough. Okay, Monica, the CAS9 collaborate a lot of company or academics side. How do you manage the collaboration?
Very well. Well, we have this joke, but it's not a joke actually, that all companies that collaborate with CAS9, they have such a great growth. They're becoming so successful, and it's based on our data because it happened to everybody who we collaborated with. So, you know, numbers are numbers. So that's definitely what we are very proud of. So, well, first of all, of course, when we decide, let's say, with whom to collaborate, we of course look, you know, what are the applications and what company is aiming for, and how much of internal resources and knowledge the company has, because then it determines how much we contribute, what is CAS9 role, and so on. But in general, you know, we have a very great team which works really super effectively, and that's the feedback that we get from all of our partners, because our Chief Scientific Officer, he has this mania about the deadline, so he always overdoes them, like does everything faster. So that's, I think, a great added value for us as a company. And through these different collaborations, we also very much learn about different applications and industries. So, you know, through these 5 years that we are in the market, we gained so much knowledge that we can actually advise our clients. So, you know, what's the best path, what's going on out there, and how they can be different, unique, and most successful. And, well, we still call ourselves, you know, a company that it's somewhere between science and business. So we are still very scientifically orientated. We have very strong scientific background, and you know, we do also publish. So that's also an important part for us, that we would create the most outstanding value. You know, really novel, innovative things. And for example, a part of our team, they are also pursuing PhDs. So, you know, the people who joined CAS9 at quite young age, they become very experienced, not only by work that they do, but also by the academic background that they are getting. So I think that's our very strong point, and also because we are really active in different countries in understanding what's going, let's say, in US, in different biohubs, and in different countries, so that then we can really offer a solution for each and every partner that we have.
Company needs money to develop their own assets. How about investment in Lithuania, investment field?
You mean what's the investment?
And your company won't get the money from the investment.
Oh, so
is it easy or?
Yeah, so in Lithuania, I think for some time we had a limitation on investments into biotech because most of the investment funds were used to the markets where they can get the return really quickly, like IT, you know, like 2 years and a company is profitable. But I think, of course, with a lot of work that was done by the biotech community, by educating the venture capitalists, so they understood that it's a different market and you know, the investment is way longer. So now we have like a boom of early stage investments, like pre-seed, seed level to new biotech companies. And when these companies are successful, then the next rounds, like Series A, they already come from investors from all around the world, Europe, US, and so on, because, well, at least the way the companies that are growing now in Lithuania, all of them are, you know, international. They work with the clients from US, from Europe, and so on. So they become attractive for investments for various of biotech funds.
Yeah, in Korea, the Toolgen is already listed in public. The COSDAQ is looks like a NASDAQ in US. And how about the Lithuania situation? What's your plan to listing your company?
So actually, this public listing, it's not very common in Lithuania, especially among the biotech sector. So it's, I think, a little different system that we have, and usually only big companies, more from traditional sectors, become listed. So that's really not a very common thing, and especially for more like a mid-sized companies.
Okay. Yeah. Now I wanted to take questions from floors. If anyone has questions, just raise your hands. No? Okay, Dr. Bungyong-gu, just tell us more specific your research field.
Yeah, so if I introduce myself, I'm a stem cell biologist working with adult stem cell based organoid system. Together with Professor Gerald Schwank in Zurich. When we were postdocs, we were correcting human patient-derived organoid with Cas9 first time in 2013, I think. So there was a milestone work that we did together a long time ago. I think that's why I was also brought into now Korea to lead the Center for Genome Engineering after Professor Jin-Soo Kim. What we are trying to do now is, because I know that Jin-Soo has started several companies, and Toolgen is also doing great research on Cas9 area. So we think that the basic institute should not compete with commercial entities and also some academic researchers who are already focusing on the already existing questions. So we are currently teaming up with people working with predicting protein foldings to understand how exactly Cas9 recognize palm sequence in a different way in different species. So I think there might be some chance of collaboration with you because you have many examples. But I currently believe that since the nature has already used insertion and deletions and substitutions to evolve Cas9 in various ways. But people in academia have been only focusing on substitution mutations using in vitro evolution. So that is something that we are currently trying to change by incorporating AI technology. And the second part that we want to focus on in the future is focusing on making genetic technology available to very distinct species. For example, South Korea is very famous for fishy farming and traditional herbs, but this technology have not been reached to that area. These are usually applied to crops and livestock. So we are thinking about developing some general delivery method to be able to amend those ones. So this is the next focus of our center. Yeah.
Okay. Any questions to the Dr. Gu? No. Okay. Yeah, time's up. Thank you for all your invaluable participation, and please let me finish this session. Thank you.
Thank you.
Thank you very much.
Thank you.